In this video, we will discuss about the mechanism of targeted protein degradation. In the previous video, we have understood the robustness of their nanoparticles that they have made that is used for targeted protein degradations, as they have tested in different cell lines and using different nanoparticle platforms. So here, the researchers wanted to find out what is the mechanisms by which this targeted protein degradation occurs. So uh, for this experiment, they have used EGFR as a control, or they have tested their mechanisms or these EGFR degradations. So the hypothesis that the mechanisms could be of these three commonly known process by which internalizations occurs within the cells, that is clathrin dependent internalizations and internalizations through this Caveolae or macropinocytosis. And so what they did is they individually block these three mechanisms using sucrose, niacin, EIPA, and then check whether this will leads to whether this will prevent the EGFR degradations or not. And they check their results using Western blot, and what they are able to see that with this 75 millimolar concentration of this sucrose, they are able to they are not able to see a decrease in this EGFR concentrations, indicating that this clathrin dependent pathway plays an important role. And similar results they have seen for nystatin, which inhibits the Caveolae dependent internalizations, and in this case also they are able to see the EGFR degradation has been prevented. However, with this EIPA, and they have tested till 5 microgram per ml, so at this concentration they are able to see some amount of the EGFR, only very few amount of EGFR compared to the case where where they have only used this NP NTZ, and on all other cases also they are able to see EGFR presence, indicating that this EIPA is not a the blocking of EIPA does not lead to the recovery of this EGFR, indicating this macropinocytosis is not the process. Sorry, macropinocytosis is not the process of the internalizations of this targeted protein degradation mechanism. So they are able to uh, confirm, or from these results they are able to see that there is the clathrin dependent and that too this Caveolae mechanisms plays a role towards internalizations of the targeted protein that are expressed on the cell surface. Thereafter, they wanted to see how this degradation occurs within the cells. So for that they target the lysosomes, and they use two different in molecular inhibitors to prevent these lysosomes, that is BAF, and chloroquine, that is CQ. And what they are able to see that with this BAF, the EGF recoveries is very good, or almost no degradations occurs, whereas in the case of chloroquine they are still able to see a degradations even with 100 micromolar concentration of chloroquine. And the recovery of this EGFR with only 100 nanomolar BAF shows, uh, with only 100 nanomolar BAF they are able to see almost complete recovery of the EGFR, indicating that it's the BAF that plays a role, but surprisingly chloroquine does not play a role. The difference between BAF and chloroquine is that BAF also inhibits autolysosomes. So they think that autolysosomes is the final destinations of these targeted proteins. So how the autolysosome works? It works by acidifications uh, of this targeted acidifications of the protein for degradations. So they again tested whether it's the autolysosomes or not by inhibiting the acidifications of the autolysosomes using using ConA. So in this case they use 10 nanomolar ConA, and they are able to see in this 10 nanomolar ConA will leads to this will prevent the EGFR degradations, indicating that is the autolysosome, or further confirming that it's the autolysosomes that plays a role towards this targeted protein degradations. Then they again confirm these autolysosomes by knocking down ATG7 gene and by silencing LC3V gene. In both the cases they are able to and both of these gene plays a role towards these autolysosomes, and sorry about that. And they are able to see in silencing these genes, or silencing LC3V or knocking down ATG7 will leads to the partial recovery of this EGFR compared to the case where there is no silencing gene. This indicates that it's the autolysosomes is the main process, and this LC3B and ATG7 genes also plays a role towards these degradations to some extent. The final mechanisms is shown in this panel G. What they are able to find out is that the internalization occurs by clathrin dependent or clathrin independent, and mainly using these Caveolae mechanisms, and thereafter it does not go to the lysosome as chloroquine. As with the use of chloroquine, they are not able to see a EGFR recovery, whereas it goes from endosomes into this autolysosome process, and that they validate using BAF and ConA. ConA by preventing the acidification, and BAF by preventing the endosomes to translocate to the autolysosomes. And they also validate these autolysosomes using ETG7 gene by knocking down ET7 gene, or by silencing LC3B genes, as they also contribute to the autolysosomes through autophagosomes. This is what they finally conclude from the results that are shown in this figure. If you need to understand more about these mechanisms or their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.